So welcome everyone to this month's IPM Speaker Series. Thanks for joining us. It's a great honor today to have someone actually speaking to us from the UK, and we're lucky to have Professor Sir Mark Caulfield. Mark is dialing in from 9 p.m. in UK time, so we are thankful. This is one of the great things about having virtual speakers is they can speak from afar, but it is a different time zone, and he was kind enough to stay up so that we could keep our 4 p.m. Speaker Series time. Professor Sir Mark Caulfield was recently appointed as the Vice Principal for Health at Queen Mary University of London, and since 2021 he has served as the Chief Executive Officer for Barts Life Sciences. And since 2008 he was served as Director of the National Institutes for Health Research Cardiovascular Biomedical Research Unit Centre at Barts. Professor Caulfield has had an illustrious career. He's published over 300 research papers, which has reaped thousands of citations. Since 1996, Professor Caulfield has administrated over £200 million in research grants. And I think one of the ones he's going to talk about today, which one of the ones is many of us feel is one of the most compelling, is the UK 100,000 Genomes Project. It was for this work that Sir Mark received a bachelor knighthood. For those that come from the UK, they'll know what that is. This is why he gets the Sir title. And it's this work that we're going to be privileged to hear today. Professor Caulfield has trained over 15 graduate students who've attracted research funding, and clearly the legacy of his work is going to continue. He has an honorary fellowship from the British Pharmacological Society, and in 2018 was awarded the Franz Volhard Award for Outstanding Science by the International Society of Hypertension. In the UK, Dr. Caulfield is chair of the Genomic Volunteers Group, chair of the National Sequencing Strategy Group, and chair of Genomics Analysis in Children's Task and Finish Group, amongst many others. He is obviously a very busy man. Goes without saying, I think we're all going to be excited to hear this. And so, Professor Sir Mark Caulfield, I'll hand that over to you. Thank you. I should say, if you want to have questions, you can put them into the Q&A or the chat, and then I will either open up the mic at the end for anyone to ask them, or I will read them out aloud if you want me to do that. Mark.
Thank you very much, Adrian, for that very kind introduction. And it's a pleasure to be with you in Pittsburgh today, even if it's virtually. If I suffer an internet outage briefly, I'll reappear at some point, but you might have to wait a moment or so. So I really have no disclosures. Between 2013 and 2020, I was chief scientist for the 100,000 Genomes Project at Genomics England, and that was a program of the former Prime Minister of the United Kingdom, David Cameron, which was designed to transform the application of genomics in healthcare. In 2019, I contributed to the development of Genome UK, Our Future Health, which is a single national strategy for genomics across the United Kingdom. It's got a particular focus in England, but now it goes across Scotland, Northern Ireland, and Wales, so all parts of the genomic landscape are reached. On the left, what you can see here is that my focus has been more recently on diagnosis and personalised medicine. That's the work of Genomics England. Genomics England is a wholly owned Department of Health and Social Care company, so it's essentially owned by the UK taxpayer. And I worked on the 100,000 Genomes Project, which is on the left. But now we've moved to adopt this in healthcare, and I'll show you how we've done that in a few moments. And what that means is there will be 500,000 whole genomes available by 2024, 25, maybe 26, that will allow us to sequence certain rare diseases and cancers in direct healthcare. We have a new cohort. It's like all of us, except it's not 1 million, it's 5 million. This is now, it was called ADD, it's now called Our Future Health, and that is just starting to potentially enroll. And then we have an asset that will have been used by many of you here, which is UK Biobank, that now has 200,000 whole genomes, but is genotyping on 500,000 and exomes on the same number. And we have an NIHR bioresource, which is up to 200,000 to 400,000 participants who are essentially enrolled against specific phenotypic criteria. And with the exception of Biobank, almost all of those can receive clinical feedback, they can be recontacted, and they are mostly UK-wide. So I'm going to focus in on the 100,000 Genomes Project and dwell for a bit on data that we published in Rare Inherited Disease in a moment. So the 100,000 Genomes project was incubated in an unusual place. Would you believe the London 2012 Olympics, where the then Prime Minister David Cameron gathered together scientists and health ministers from around the world during the Olympics, and they had a summit in which he was advised the moment was right for a free at point of care health system to adopt whole genome sequencing. And as a result, small groups were convened. And when I joined in July 2013, the 65th birthday of the National Health Service, we had 4 letters, one on rare disease, one on cancer, one on infection, and one on informatics. And these were 3 sides of A4, and that essentially was the blueprint for the program, which my colleagues and I at Genomics England had to create into a formal protocol and bring live. What was very clear was that this needed to be tractable to healthcare benefits to have a direct relevance for the health system and to be adopted at the end of the program. So to that end, we focused on rare inherited diseases, cancer and infection, because there were tractable benefits that were evident then. We've reached over 97,000 people, patients and family members. We've over 40 petabytes of data. And to do this, we needed it to be really owned by the frontline healthcare system. And so with the National Health Service, we created 13 genomic medicine centres of excellence across England, and then involved Scotland, Northern Ireland and Wales. That reached 98 of the 220 NHS trusts, and NHS trusts being a group of hospitals that serve a community. But what it means is there was regional equity of access to this programme, irrespective of ability to pay. At peak, and this is really, really important, 5,000 frontline NHS staff, doctors, nurses, other allied health professionals and counsellors were involved in this programme. And that meant that at the end of the project, we hadn't marched a group of people to the top of the hill and abandoned them. This group of people were ready to be the transformers in healthcare. And that has brought live a new genomic medicine service. Now, as anyone here listening will know, because this data is not fully worked out, we openly advertised in Nature on 2 occasions, and anyone, including any of you listening here today, can join the Genomics England Clinical Interpretation Partnership. This is an international research collaborative of over 3,000 researchers from 33 countries in the world, and they work on the data for free to drive up its value for clinical care. So I'm going to move to rare inherited diseases. In Western societies, typically these affect about 6% of the population. I'm going to focus on a pilot study we published in the New England Journal in November 2021. This reached 4,660 participants, and it really was the proving ground that we could actually do this. And they're from 2,183 families, and they have 161 disorders. We used this to establish standardized eligibility in phenotyping. And we used the human phenotyping ontology so that if you used it in Pittsburgh, you would be able to access my data and use the same terms to search across my data to find people with analogous phenotypes. We automated the analytics, and we involved the NHS in confirming initially the gene panels we looked at, because we did look at virtual gene panels, but then we actually looked at genome-wide analysis as we were moved on. So what you see at the bottom is the different number of families recruited in the main program, where we have 36,000 families with rare disease and a 20% diagnostic yield. But I'm going to focus today a little bit on the pilot, because there's some important learnings for anyone who's involved in the healthcare system in this respect. So the picture on the left-hand side is a family who really want you to see them, because their daughter in the middle there was 4 years old when she was enrolled in the program, and she presented with falling behind her milestones and against her peers, and then she developed what was evident intellectual disability and intractable seizures, unresponsive to any anti-epileptic. We found in our whole genome a glucose transporter 1 mutation that had previously been missed by exome sequencing and other orthogonal testing. And as a result, we discovered by analyzing her family alongside her, and parent-offspring trios are very powerful in this respect, as I'll show you in a moment, that this was a de novo mutation in her, and her parents didn't have it. They'd not been going to have any further children, but as a result, they are now trying. And so this not only paved the way for a genomic diagnosis for this young girl, but it also was transformative in that you can treat glucose transporter 1 mutations, which effectively interfere with sugar transport or glucose transport from the blood into the brain. You can treat it with a high fat or ketogenic diet, and that's what we did with this girl. And she now makes her own sugar in her brain using a starvation mechanism all of us have. And her seizures have greatly reduced and her intellectual disabilities improved. What if you and I could have discovered this shortly after birth or early in this child's life? What would be the influence of that on her outcome? Here's the journey of a genome. So you're all billionaires here. You've 3.3 billion letters in your genome, and you spend that genome over your life course. In this child's case, she had 6.4 million variants. That's slightly more than usual. 677,000 were rare. 2,826 were predicted by bioinformatics to change a protein. 67 were different from a mum and dad, and 1 was a clear-cut cause of her condition. So that's what we do in a matter of hours every day with the families analysed in phase so that we can take advantage of the mums and dads' genome to help us filter down the variants. So let's look at some clinical stories from this program. So in the New England Journal, you'll see that we published several cases, one of which is a 4-month-old with fluctuating neurology who died with no diagnosis. His immune testing was negative. He was enrolled in the project by his parents. His mother became unexpectedly pregnant, didn't want to know the diagnosis. We found she became very anxious at month 8 and said, actually, I would quite like to know. So we found a pathogenic transcobalamin 2 mutation, which meant that when his brother was born, he could be tested within 1 week. And sadly, we discovered he was affected with the same mutation. But because of case reports dating back to the 1970s, we administered high-dose weekly vitamin B12 injections, and that child has had a totally different course from his elder brother. The second case, a 10-year-old, a girl admitted to ITU on multiple occasions with life-threatening infections, on this occasion chickenpox. And she'd had detailed immune testing and genomic testing with no diagnosis for a presumed immunodeficiency. We found mutations in CTP synthase 1 that affects B and T lymphocyte responses to infection are both capsulated and bacterial infection. And so this child was highly susceptible to severe infection. As a result of this diagnosis, she was listed for a bone marrow transplant, which he's had, and it's curative, the impact of which is dramatic. So this child cost the NHS £357,000 in multiple admissions to ITU. For just a few $100s, we did a whole genome, and for 70,000 sterling, probably about $85,000, we did a curative bone marrow transplant. But when you compare that to £357,000, doesn't sound that much. The other value is that we were able to test her siblings and to reassure her parents that these were not at risk of infections. And now if you look at this girl's history, instead of being multiply admitted to hospital and tripping often to the ITU, she actually only comes to outpatients for monitoring of her bone marrow transplant. And the final case is a child that had, this is an example of where a whole genome can reprofile a diagnosis. This is a 5-year-old with unexplained anemia, developmental delay, short stature and constipation. He'd had a diagnosis of diamond blackfan anemia that may have a limited lifespan, potentially with cancer risk. But because of the developmental delay, which was a little atypical, we deployed an intellectual disability panel to analyze the genome and found a tier 1, in other words, a pathogenic but de novo variant in him, in thyroid hormone receptor alpha, which means that although his thyroid function is ostensibly normal, he cannot respond properly to his endogenous thyroid hormone. But if you replace him with high-dose thyroid hormone, it can be transformative for him. So this child is quite different. And you can see this child's video. He's called Owen, and his mum and dad would like you to know his name, on our website. And you'll see that initially in the video, he walks hand in hand with his parent and has a rather unsteady gait, but by the end, he's dancing. And this child is now a different person. What if we could have discovered that and these other diagnoses much earlier? Here's the words of a mother in the program. This is a 5-year-old with rare disease. This child turned out to have GAMP deficiency. And at 5 years old, these are the words of a mother, which I'll let you read. But essentially, her principal concern was her daughter was showing degenerative neurological change, and she was waiting for her to end in a wheelchair. She got stuck at about a developmental age of 12 months, became very much locked inside with no emotion, unable to communicate. But 6 months after a simple diet shift with creatine, ornithine, and arginine, this is a new child with light back in her eyes. And this lady stood up at a conference and recited this. This was a very emotional experience for everyone who was there. But for those of you who face people with rare inherited diseases, when you make a diagnosis, even if you haven't got a treatment, the elation of that family for the end of a diagnostic odyssey and uncertainty is huge and a massive thing. So here are the data from the New England paper. So these are the demographics of the 2,183 families. You'll also see that the families had a higher higher age, and there were more adults in this program than you might normally expect. This reflected pent-up demand with unmet needs. So the longest diagnostic odyssey in this program is 30 years. So, you know, these are people who went through the health system, had every test that was available, but didn't actually get a diagnosis. And so in the main program, there are many more children. The age is younger, but we also have parents, and therefore we can cover both adult and pediatric rare diseases. So here is the impact of diagnostic yield. So we, in the New England Journal paper, reported a 25% diagnostic yield. Here you can see in the middle the impact on diagnostic yield of having just a single family member that's affected versus a duo versus a trio. And as you get larger, typically line of sight on diagnosis improves. The reason that quads and larger family structures are different is because trios, family quads, and larger family structures are populated by some of the more challenging diagnoses. So this is a sort of an artifact. But by and large, the central observation is the more family members you have, the better your line of sight and answer. On the right, I'd like you just to look briefly at what the difference was if you partition these disorders into those with a likely monogenic etiology or those who were likely to have a more complex etiology, probably oligogenic. So what you can see there is that with research added in, you can add up to a 37% diagnostic yield and an approximately 11 to 12% diagnostic yield for oligogenic disorders. Not in the paper, but nonetheless important, is in the blue in the pie chart that you see on the left is the splice site SNVs indels that you might have captured in the coding region in panels or possibly exomes. In the orange, green, and yellow is the added value in terms of variation that sits outside those panels in these families. So you can see that a number of the diagnoses, 14% approximately, came from outside the exome. And I think as our knowledge increases, that will become more common. Typically, these are intronic regulatory regions, splice junction mutations that are not easily measured by an exome because it tends to truncate at the splice junction, or even things that sit at some distance from the gene that you and I might not have suspected. Another phenomenon we encountered on a number of occasions is a situation of a compound heterozygote where there is a rare variant that essentially is effectively a knockout variant that knocks out 50% of the protein expression, and then a common variant that's at 20-30% frequency that alters as a missense a 5% or less, and that's enough to tip people over into disease. They present later, they obviously survive longer, but they also get the disease because they've just lost too much on the critical path to the disease from that protein. Here you can see a program invented by my friend and colleague Damien Smedley called Exomizer, developed with Peter Robinson at the Jackson Labs as part of the International Mouse Phenotyping Consortium. And this combines multiple phenotypic information from multiple species with humans. And what it does is it up-prioritizes signals that are more likely to be true positives. And Exomiser made about a 15% augmentation of diagnoses when we deployed it here in the New England paper. I'd encourage anyone to adopt Exomiser to prioritize signals if you're not using it already. It's free on the web, open source software. Here from the New England paper are the absolute numbers of families with different disorders and the diagnostic yield according to category. And what you can see here is that we're showing you the diagnostic yield, and then what you can see here is the difference that prior testing makes. So whether you have prior testing or no prior testing, you get about a 30% uplift in diagnostic yield. And that's true whether it's chromosomal targeted NGS panel or WES, although I should caveat the whole exome sequencing here, in as much as there's only 16 people in this, and therefore one needs to be cautious in interpretation. So just to take you on the tree of outcomes in this program, there were 2,183 probands, 25% had positive reports on first analysis, 535. So we got diagnosis based on coding SNVs or indels or in the applied panels. If there weren't, then they went on to research, and a further augmentation in diagnosis was achieved by review by two companies, Congenica and Fabric. And if that wasn't successful, they went on to research phase, and additional diagnoses were based on coding or indels or mitochondrial DNA or various regions of the genome. And then there were further reports, as you can see here. And we had 10% at the end that had variants of uncertain significance. Approximately 65% had entirely negative reports at the initial stages after proper research. We're gradually eroding that. So with time, new knowledge, better improved algorithms, we can augment this by between 6 and 8%. So a genome is a relatively static but life course beneficial analytic, and it is very useful for ongoing care. I'll just take you through the diagnostic odyssey of children in this program, born after 2003. So they spent a median of 68 hospital appointments, 75 months, approximately 6 years, attending appointments not to get a diagnosis. And then after they had achieved diagnosis, they had fewer focused episodes. But most importantly, let's contrast that with their unaffected siblings who attended 18 appointments over 120 months, a much longer period, almost double the period. The affected participants in this program used 183,273 episodes at a cost of 87 million as children, and a median cost of £15,310 per participant. For a few $100s, you can do a genome. This is suddenly cost effective. Because this is lost in insurance programs or in direct healthcare over a number of years, this figure does not come to light, but that's nonetheless what they cost. Now, it's not to say that their unaffected relatives who have accidents, infection, do not have hospital contacts, but they spend far less. So this doesn't include family physician visits or disease treatment costs. So let's look at the healthcare benefits. About 25% of these diagnoses had an immediate utility. Some of them it changed medicine, some of it led to additional surveillance, some were eligible for trials, for some it informed future reproductive choices, others benefited from dietary change or vitamins, and there were 829 genome-wide significance, 249 were in known genes for rare disease, 579 are potential diagnoses, but at the moment we haven't got absolute proof for more than 22 of those, which we've done further work on. So I'm emphasizing here to you today that the barrier is not our ability to interrogate the genome, it's about making functional sense of what we find there and converting that into a real diagnosis that provides the biological insight you and I want to have to transform healthcare. Now, the totality of that pilot, it cost approximately 237,000 episodes of care, £106 million. That's $138 million. Here's something we published in February 22 in Lancet Neurology. So what was thought when we started in 2013 is that you couldn't really use short read whole genome sequencing from Illumina at approximately 150 base pairs to diagnose repeat expansion disorders. And so we looked at the 13 common known repeat expansion disorders and used and applied an algorithm called Expansion Hunter, which I would encourage any of you to adopt in the health system. So what you're seeing here is in the blue, a histogram of repeat length by different types of repeat expansion disorders. So you've got ataxin 1 to 7 on the left, on the right-hand side, and Huntington's disease at the bottom. What you notice is there is a red serrated line to the right of the blue histogram. That denotes the repeat expansion boundary beyond which a repeat measured as expanded is pathogenic and usually leads to neurological disease. These are the most common neurological disorders and affect 1 in 3,000 of us. And what we're showing here is that these were all people enrolled in the early phase of the 100,000 Genomes Project who had no diagnoses, many of whom had hereditary ataxias, and you can see that many of them are receiving a diagnosis from a whole genome. And what you can see in the red triangles are repeat expansion disordered participants. And I'm going to draw your attention to the Huntington's disease row because this is important. If you look at that, immediately to the right of 40 base pair repeat size is a red triangle. And then to the far right, just below 100 repeat size expansion, is a shaded red triangle. This shaded red triangle is a child that's under 10 years of age that is presented with an atypical presentation of Huntington's disease, but has a massively repeat expanded region in their genome. Their dad didn't have any symptoms at that time. He's the person next to the red serrated line who has a repeat expansion disorder but had not manifest any features. And this is often the case that dad has transmitted with a substantial further repeat expansion to his son, the trinucleotide repeat. You can measure this with whole genomes. We have validated this against orthogonal PCR testing for known cases, and this is now ready and incorporated in the NHS into routine diagnosis. We're also running this to identify new potential repeat expansion disorders, and this is the work of one of my fellows, Ariana Tucci and Christine Ibanez-Garicana. I'm going to move to cancer now because cancer was also in the program. So we worked on 17,339 cancer participants. And these cancer participants had their tumor sequenced because their cancer is a disease of disordered genomes, and also their germline, usually from the white cells in their blood. We made every attempt to optimize formalin-fixed paraffin-extracted tissue, but whatever we did not work. Here is evidence of that. What you're seeing arranged in a circus plot, in other words, a genome round in a circle from chromosome 1 to X, is that you can see a gentleman's genome with prostate cancer. It's the same man's genome on the left and the right. The difference is on the left, the genome is provided from fresh tissue that was freshly biopsied from the same cancer as the genome on the right. The difference on the right is that that was exposed to formalin fixation. So if you look at the genome on the right, there are several notable things. One is that the genome is quite moth-eaten and has a sort of an irregular outline. In the middle, you can see lots of green crosslinks. This is spurious formalin cross-linking. You see on the left-hand side, there are far less structural changes, but on the right-hand side, you've many spurious changes. On the right is a product you can't use in healthcare. On the left is something you can rely on. But if you go down to the metrics, just look at the FFPE versus fresh frozen tissue for SMV calling and indels. A dramatic expansion of false positive SMVs and indels. You can't use this in healthcare. So we re-engineered 400 molecular tissue pathways and used those to generate 17,339 patients, the largest single cancer repository in the world of fresh tissue whole genomes. And I would encourage everybody looking at this not to use formalin fixation. What can I say about other alternative fixatives? I can say that alcohol fixatives work pretty much as well as fresh tissue. So if you need to use a fixative, though we do not know the longevity of the preservation of the tissue, you can use that. So let me give you an individual case. Let's bring it back to an individual. This is a man who was in his 40s, well-controlled HIV patient, previous hepatitis B, presented with weight loss and jaundice. On the CT scan, which essentially is a cross-section through his liver, the liver is a grey area on the right of your picture. And what you'll be able to see is that there's a denser grey region in the center. That is tumor replacing the liver. And it turned out to be adenocarcinoma from colorectal origin. We did various tests, usual care testing, found nothing that would influence his care. He had 6 cycles of usual chemo, got worse. He was rebiopsied. We found that he had mismatch repair disorder, in other words, Lynch syndrome, with an MSH6 mutation, and he got prioritized for immunotherapy trials. Now, sadly, he's not going to survive this, but this is a missed case of Lynch syndrome presenting late. And what it does say is there are certain things which, if we were able to screen for them and could identify them earlier, particularly in a family history of early onset colon cancer, maybe we should think carefully about that. What if we could have prevented this? I'm going to give you an example that we've recently discovered, which is effectively the use of a whole genome in carcinoma of unknown primary. Now, the problem with carcinoma of unknown primary is when it turns up, you're never really entirely sure what the best treatment for the patient is, and typically the outcome is very bad. So this was what looked like a metastatic deposit in the brain. It wasn't a brain primary, it didn't have the architecture of glioma. So we started to look at the molecular signature from the whole genome. You can also, by the way, do this with epigenetics. As you may know, epigenetics may even be a more reliable indicator of tissue of origin. So we found TP53 mutations. This could be relevant for many cancers, as you know. We found increased tumor mutational burden, which you also see in association with lung cancer, and that narrows it to colorectal, lung, and melanoma. And if it was a woman, this is a man, it would be endometrial too. Here we see a benzopyrene signature, which is a smoker mutation signature in the tumor. So this was almost certainly lung cancer. This allows tailored cancer care. So this is a genome telling us the likely organ of origin and tailoring therapy. So this was never a no-hope result in cancer because of 3,244 patients in the West Midlands Centre, which is in the middle of England, 477, 61% were returned for a clinical evaluation by a genomic tumor board. 53 had mutations that affect metabolism of 5-fluorouracil and capecitabine such that you could be exposed to severe toxicity. 21 had a clinical action with a licensed drug they wouldn't have received. 11 had an unlicensed drug they wouldn't have received. 204 got into a clinical trial they wouldn't have got into. And 43 had high tumor mutational burden, which may direct you to early immunotherapy. And 69 could be referred to clinical genetics. So that's 25% approximately gained a benefit from their cancer for their cancer care. So this is not a no-hope result. Here's something that we started to do with machine learning, which is to use routinely collected cancer registry patient data. Every hospital in the United Kingdom submits data to a national cancer registry, including pathology reports. So we use machine learning to go in, extract staging information and various other key points of data that can be organized and curated into a research-ready data asset. And we've used that real-world data to demonstrate this. Now, this is not a new finding. This is where what we've done is to look at the impact of the somatic variant on PIK3CA, the wild type, which is the black line. The wild type is the pink line to the bottom there on the Kaplan-Meier survival curve. And what you can see is over time, a separation between the wild type in pink and the black mutant. Black mutant is longer survival, which is protective. And this is generated just like in a clinical trial, but from real world healthcare data. And in 2012, this was reported by Bichet and colleagues in breast cancer research. So it's not new, but this is real world data. So you can do that if, as we do, longitudinally life course follow everyone. Here's something else that we've done. We've looked at 76,000 whole genomes, to look for known clinical pharmacogenetics international consortium gene-drug pairs. And what we find is, irrespective of ancestry, 99.5% of us have haplotypes that are potentially harmful in at least one gene-drug pair. So in one known pharmacogenomic gene. So what that means is we either will be at risk of harm or inefficacy of the therapy. And remarkably, 25.4% of us have at least 4 of these. So you may have seen in the UK, the Royal College of Physicians and the British Pharmacological Society have just published a report, which I co-authored with others, on pharmacogenomics and the time for adoption in healthcare. And you have a bill passing through US Congress to adopt pharmacogenomic response mode testing, but this will move to preemptive testing. And you can get this information for 53 gene-drug pairs reaching about 80 drugs out of a whole genome. It's a byproduct of having the whole genome. What I'm seeking to emphasize is a genome has life course benefit, but it also may have benefits beyond the primary reason that you enrolled the person. Here's what we did to generate research on these data assets. So we openly advertised for researchers to volunteer their time and their skill set. And in blue, you can see the countries from around the world where people have volunteered from. And 3,580 researchers have volunteered. About 3,000 have access from 413 institutions in 33 countries. And they've won for themselves £50 million in grant. They have a 43% grant success rate, and they have access to 4 billion plus clinical data points and over 111,000 whole genomes. We also have a partnership with companies, adding value to the data. And here's what we've done with this data. We've used this to create a new national genomic medicine service, which offers regional equity of access to care for the whole of England. And to do that, we created 7 genomic lab hubs. These are regional lab hubs. They do all testing that's not a whole genome. And they do it according to a national test directory. My team and I, when I was at Genomics England, reprofiled 300,000 tests and upgraded 25% to new technologies. And what we're doing now is consenting for 500,000 whole genomes that will add to the 100,000 genomes and create a national UK genomic knowledge base. And where we have consent, that data will be made available for research. And Genomics England continues to oversee our whole genome sequencing platform, which is a NovaSeq platform, which is based in the Sanger Centre in Hingston, but run by Illumina on our behalf. In addition, to manage the interface with patients, we've created 7 genomic medicine service alliances, and we have a workforce development program and a national masters and other fellowships that are being delivered to build out capacity in the health system. Here's some of our most recent work, which we published in Nature in February 2022. So when COVID struck, we were asked to pivot and face our program towards COVID. And so we organized whole genome sequencing in severely COVID-19 patients, 7,491 from 224 United Kingdom intensive care units across the NHS. We compared those genomes with 48,400 unrelated controls from the 100,000 Genomes Project and validated our findings in the Host Genetics Initiative, minus the genetics of mortality and critical care study. We also undertook an in silico TWAS and Mendelian randomization and gene burden testing. So what did we find? We found that you can adjust the p-value for a whole genome. So if you adjust it to 2.2 times 10 to the minus 8, you can correct for 2.26 million independent tests. 23 of these signals were genome-wide significant, and we identified 16 novel loci. And in trans-ethnic analysis, we identified 3 signals that passed genome-wide significance. This signal is mostly driven by South Indian Asians, but individually no ancestry crosses genome-wide significance. And the 3 gene regions include TRIM46, BCL1A, LINCO1276, and TWAS shows multiple significant fold changes in expression. Gene burden testing showed very little, but we do have 1 rare variant, which I'll comment on in a moment, and 2 putative therapeutic targets. One is mucin-1, which is associated with excess mucus production in the lung, an inflammatory consequence of COVID-19, and potentially causing lung damage. That can be potentially blocked. and then colony stimulating factor, which can be interfered with by GCSF or provided by replacement by GCSF. Here are the loci. It says 25 there, it should be 23. And what you can also see is that in the middle on chromosome 8 is interferon alpha 10, and that particular gene locus is dependent on a rare variant, which was only replicated and validated with the help of Regeneron's data. What you see here is the importance of these loci. They categorize into roughly 3 or 4 categories. One is inflammatory gene regions, immune gene regions, clotting gene regions, and ABO secretor blood groups. And the HLA-DRB1 region is positive in our data, but we've not been able to replicate that because most replication resources are based on genotyping, and often they poorly cover the HLA region. So I'm going to move finally to genomic analysis in children. And the task and finish group Adrian referred to when he introduced me. I was asked by the Chief Medical Officer of the United Kingdom to look at whether we could evaluate the role of next generation sequencing. And in 2016, she published a report, this is Sally Davis, called Generation Genome. And she was unhappy with that report because for newborn screening, it had recommended a very slow progression to panel testing from the 9-point mass spec measured Guthrie spot that we use today. Even in the US, you already measure more things in newborn screening. So she asked me to look at 2 areas, intensive care and newborn screening. So I formed a group of broad multidisciplinary expertise, including patient advocates, patients themselves, and broad representation of the community. As a result, we brought a rapid exome service live in October 2019 with 37% or 39%, depending which year you look at it, diagnostic yield. We have identified new treatments for some of these patients, and a number of patients that came in during COVID for rapid exome sequencing with children have received an immunodeficiency diagnosis they wouldn't have received. So at the moment, we have exome sequencing live, but like you in Pittsburgh, we're looking also at the potential of whole genomes and accelerating that turnaround. So the recommendation of this report was that whole genome sequencing should be deployed for all children in NICU and PICU with no clear diagnosis who have been admitted there, because the evidence from programs across the United States is that you can increase diagnostic yield by deployment of these tests to between 30 and 51%, and that you can also alter the care pathway for those patients. Sometimes it will be a treatment, but sometimes it will be palliative care. In newborn screening, the group considered how we might approach this with the delicacy that this involves, and recommended very extensive societal and ethical consultation. As a result, we looked at what we would do with this, and we concluded that the production of raw sequence was the assay, the measurement, that you could analyze this assay at any given context, clinical context, at any different point, and that was the analysis. And then the return of results by consent was the output. And these elements could be temporally segmented or separated, and this could allow us to address the use of a genome or longitudinal life course of an individual. We recommended ethical and societal debate before, during, and after the program. And the first thing we considered was, should we use the whole genomes? And we concluded we probably should in a test versus the standard newborn blood spot and the associated 9-point analysis. So usual care versus a whole genome. And ask the question, what extra health or technical benefit might there be? Could we see novel uses not currently employed in newborn screening? We would need a direct comparison of the yield. We would need to know what the cost of this assay was, and we would also need to be able to have a national debate.
So, first thing we realized was that there was no single version of the truth for what constituted an archive of genetic disorders that were treatable. So working with David Bick, then at Hudson Alpha, now at Genomics England, and Richard Scott and Tom Fowler, we established the website RxGenes, with David Dimmock's help too. RxGenes is available as an app from the App Store, and you can download it right now. and plug in any disorder of your interest, and it will tell you whether it's a treatable disorder or not. So we then looked at how you could use this in the field, and here's an example of the app in action with phenylketonuria, advocating a low-protein diet and other things that might help with the treatment of this disorder. We then looked at how many disease genes would be treatable, and we discovered there were 681 disease entities that mapped to 665 genes. This is because 13 genes result in 2 or more diseases with variations in different parts of those genes. 20 genes were associated with adult disorders, and 4,525 genes were based on phenotype causing variants listed in Omen. So we then started to look if these were fully penetrant disorders, and for fully penetrant disorders, we found 639 disorders, an estimated 1 in 190 live births. Now, given that there are 712,000 live births in the UK in 2019, that's circa 10 children born every day. Taking the same 2019 figure of 3.75 million for the US, that will be 54 children born every day. So right now, we're running a sequencing pilot to investigate the false positive rate. This is the number of times we will claim a variant is associated with a potential disease risk, and that won't be true. And so we're using a cohort called Born in Bradford that was enrolled very early in the last decade, at the beginning of this decade, and that will allow us to see variants that we think are potential true positives, but where the phenotype has not been manifest. The benefits of this could be substantial. Our estimates suggest that funds released from some of the more severe disorders could range between 360,000 and 1.4 million sterling for an individual. The quality adjusted life year gains are also substantial, and this is because this affects children born with disorders that present before their 5th birthday. So you can gain in QALYs 16.8 to 40.3 years. And that means also, if you look at the cost effectiveness, you can achieve an incremental cost effectiveness ratio of less than 50,000 sterling, which could be considered cost effective. We then went on to test this in the public and participants in focus groups. And there was overwhelming enthusiasm for this, particularly if it was representative of the UK, generalizable across centres, not just centres of excellence and ivory towers. And if we robustly tested usual care versus a genome and used transparent processes to communicate this to society and to lay the foundations for a technology shift. So now, the new programme of Genomics England, which accords with Genome UK, our national genomic strategy in the United Kingdom, which is available on the web, focuses on diversity, genomics and pharmacogenomics, a new cancer programme, and newborns, where we are looking at the programme I've just described. We'll also continue to work on rare diseases. So, ladies and gentlemen, we've used this information to transform genomic medicine in our National Health Service. So now, as a result of the 100,000 Genomes Project and the matters I've covered in this talk, there is a National Genomic Medicine Service providing equitable care for 56 million people, standardized, with a standardized common genomic consent for both NHS care and research, delivering against an approved national test directory, which is the basis for reimbursement. So this is a test directory against which you receive payment for the test done that ranges from single gene to whole genomes, because today you can't make the case for universal whole genomes. We're building a single UK knowledge base, and we'll have a national database that will be available de-identified for academia industry. But I've done this talk this evening because, like you in Pittsburgh and those who are watching this through the web, the future is a global coalition of intellects that drive genomics into healthcare. And our goal is for the UK to be at the heart of that, but we hope you'll all join in. Thank you so much for your attention this evening. And I'd like to thank our most important people in this program, which is not me, it's our participants. And then I'd like to thank the colleagues who worked with me on the New England Journal paper. Quite a large number of people there contributed to that. And this is their acknowledgements. And I'd also like to acknowledge the colleagues on the COVID program and the families and participants across 509 ITUs, and encourage you to stay in touch. Please do get in touch if you want to know more. Thank you so much for having me here